This video is from the Chemistry of Life unit, Biochemistry and Enzymes, Metabolism, Chemical Reactions. Let's first draw a graph. This graph is going to be an energy graph over reaction progress. So on the y-axis is energy, and on the x-axis, reaction progress. What we can do with this graph is show how reactions change in the presence of something called an enzyme. So if our reactant started out with this much energy, without the presence of any chemical or catalyst, there would need to be an input of energy in order for the energy to go and in the reaction to go and form products. This energy here between the reactants and when the reaction gets going to products is called the activation energy. In order to reduce the activation energy so that reactions can go faster and without as much energy, the activation energy needs to be lowered. And this happens in the presence of an enzyme. An enzyme is a biological catalyst. An enzyme that is a catalyst is something that speeds up a reaction. The way it does that is by reducing activation energy. An enzyme reduces the activation energy by putting substrate in proximity. An enzyme is a globular protein. Enzymes have specific shape. This is a schematic drawing of an enzyme. What makes this drawing like an enzyme is that its shape is unique to its substrate. This globular 3D protein structure can bind with a specific substrate. This substrate that I'm drawing right now would have to be the exact same chemical shape. More than one substrate can bond to an enzyme. These substrates are the reactants in the reaction. These substrates are positioned so closely to one another that they might form bonds and stick together. The reason they're in such close proximity is because of this area of the enzyme. That area of the enzyme is called the active site. And the active site is so specific to substrate that it's like a lock and a key. As the reaction goes through, what results is a product. This product is now formed from the two substrates that combined, and the enzyme remains exactly the same. It is unused. Because the enzyme is unused, unchanged, and unaltered, it can be used over and over again for multiple reactions. Enzymes are found in all metabolic reactions in the body. For the different kinds of macromolecules that we discussed in the Chemistry of Life unit, there are some chemical tests that we performed in order to identify the presence or absence of these macromolecules. To test for carbohydrates, we did two kinds of tests. The first one was the Benedict's reagent test. Benedict's tested for reducing sugars, and this turned orange in the presence of monosaccharides or disaccharides. The other kind of carbohydrate we tested for was polysaccharides, and we did so by using an iodine test. Iodine goes from amber to black, and the specific polysaccharide that we were testing for is starch. When testing for proteins, we did a burette test, and it turned violet if protein was present in the solution we were testing. And when we tested for fats, the teacher did a Sudan 4 test, which turned red, or a translucence test on brown paper, where the brown paper turned slightly see-through in the presence of fats. That's the metabolism chemical reactions section from the Biochemistry Enzymes review in the Chemistry of Life unit.